Hello everyone, welcome back to my channel. I'm back with one more video. How are you doing? Hope everyone is fine out there. So let's get into the class. So in the last class I have already discussed about the pyruvate formation from glycolysis and also the fate of pyruvate and the enzymes involved in the glycolysis. Okay, based on the presence of oxygen available, the pyruvate may either enter into anaerobic respiration or the aerobic respiration. In today's class, I want to discuss about the most important central metabolic pathway that happens in all the cells is the citric acid cycle. Okay, so this citric acid cycle name comes from the citrate, which is the first product formed in citric acid cycle. Okay, this is also named as the tricarboxylic acid cycle or the Krebs cycle, which is named after a scientist known as Hans Krebs, who discovered this particular cycle. Okay, so why this citric acid cycle is called the central metabolic pathway? Because this citric acid cycle produces all important raw materials that are meant for the other amino acids production also. So all the metabolisms of fatty acid metabolism, the carbohydrate metabolism and the amino acid metabolism finally enter this citric acid cycle. Okay, this citric acid cycle plays an important role for the production of NADH, FADH, GTP, which are meant for the conversion into ATP in the electron transport chain. In this class, I want to concentrate mainly on the citric acid cycle, okay, which is most important cycle that happens in all the eukaryotes and in the prokaryotes. Okay. Now, so it's a biochemical pathway that is also known as TCA. I told you it's a key metabolic pathway that connects the proteins, fats and carbohydrates. Okay, and this cycle generates energy through the oxidation of acetyl-CoA. Okay, you all know that oxidation and reduction plays a very important role in this cycle because all the enzymes involved are meant for the transfer of electrons. Okay, I will tell you. Okay, let's talk about the location. So before I told you this glycolysis happens in the cytoplasm of all the cells, but prokaryotes doesn't have a proper mitochondrial formed. So here the Krebs cycle happens in the cytoplasm of prokaryotes, but in eukaryotes this particular cycle happens in the mitochondrial matrix because mitochondrial matrix is nearer to the membrane of your mitochondria, inner membrane of your mitochondria, right? In the last class I told you about the structure of mitochondria. The inner membrane is folded in the form of cristae and all, and you also happen to hear about the F1 particles which are meant for the electron transport. Okay, so as this Krebs cycle happens nearer to electron transport, this pathway is considered to be the central metabolic pathway. So that's why all the metabolisms enter into this Krebs cycle for the conversion into ATP. Okay, so now let's discuss about it. So in the end of glycolysis, you have a net gain of 2 molecules of pyruvate and 2 molecules of ATP for 1 molecule of glucose, right? And this pyruvate, what happens, now gets converted into acetyl-CoA. So this acetyl-CoA with the enzyme called pyruvate dehydrogenase. So pyruvate dehydrogenase are a group of complex of enzymes where the conversion happens, where the hydrogen is released from one molecule, making it an oxidized one, and this transfer of hydrogen would happen to the other molecule, making it a reduced one. So that is what you call oxidation reduction reactions, and these reactions are mainly catalyzed by enzymes dehydrogenases, right? Okay, this you happen to hear a lot in this Krebs cycle because so the 2 molecules of pyruvate is acted upon by pyruvate dehydrogenase complex that is added with a coenzyme. This coenzyme is coming from a vitamin B5. Okay, that would help for the conversion. Okay, the 2 carbons are mainly transported from the pyruvate to coenzyme A, forming the acetyl coenzyme A. Okay, here one molecule of NADH is produced, like NAD plus gets reduced to NADH. Okay, so how does the Krebs cycle produce energy? So why is Krebs cycle very important? Because I told you, because of its location, because the Krebs cycle happens in the mitochondrial matrix, which is nearer to the membrane of your mitochondria. This is considered to be the central metabolic pathway. All the metabolites would enter into pyruvate, into Krebs cycle, so that they could be easily enter into the electron transport. All FADH and NADH molecules would easily enter into the mitochondrial membrane for the electron transport to happen. So that is why Krebs cycle is considered to be the important cycle. Okay, and now I told you after after the conversion of one molecule of glucose to pyruvate, there is only net gain of 2 ATP, right? But at the end of Krebs cycle, or at the end of electron transport, when they enter into electron transport, the complete molecule, one complete glucose molecule needs to the formation of 36 number of ATP molecules. Okay, after all the pathways, the net gain would be 36 ATP. Now I will discuss the electron transport in detail in the next class. Okay, in this class I am concentrating only on the Krebs cycle. Okay, so apart from glycolysis, apart from glucose metabolism where glucose is broken down, there is another reaction happening where the glucose is converted into a 2 carbon acetate. Okay, this 2 carbon acetate is, after a series of reactions, would be released in the form of 2 molecules of carbon dioxide. The 2 carbon compound acetate releases into 2 carbon dioxide molecules at the end of your Krebs cycle as a waste product. Okay, that is how you will get energy for the Krebs cycle. Okay, and apart from this, you will also hear about the transfer of electrons. I told you the NAD, there is a transfer of electrons from NAD producing NADH. In the same way, the electrons are added to FAD to produce FADH. So in these are there is also a conversion of GDP to GTP, which is you can say it's a cousin of ATP molecules, where the same reaction happens where the inorganic phosphate get transferred, and thus you will be producing GTP also in the end of Krebs cycle. Okay, let's all discuss in detail about the Krebs cycle in the next slide. Here comes the cycle of Krebs cycle, where it starts with acetyl-CoA. This acetyl-CoA is a 2 carbon compound which combines with the 4 carbon compound called oxaloacetate, producing citrate. Okay, this enzyme called citrate synthase helpful for the conversion into a 6 carbon compound citrate. Okay, here one molecule of hydrogen is used. Okay, I will tell you in the next slide what is the net gain and the result of Krebs cycle. Okay, and this citrate is again converted into isocitrate, which is an isomer, and isocitrate being more stable than citrate, this isocitrate is produced with enzyme called aconitase. Okay, there are completely 8 to 9 enzymes involved, and you have to remember all the enzymes for your exam purpose. Okay, and here the conversion of one acetyl-CoA, which is a 2 carbon compound, leads to the 2 carbons in the form of carbon dioxide. Whatever the acetyl-CoA that is added here would be released as waste product in the form of CO2. Okay, in this step and in this step I will tell you. So this isocitrate is converted into alpha ketoglutarate, where NAD plus is reduced to NADH, and it is synthesized, it is catalyzed by the enzyme called isocitrate dehydrogenase. So you know that dehydrogenation is mainly catalyze the reaction of oxidoreduction reactions, conversion of hydrogen atom, oxidizing it, and converting transferring of hydrogen atom getting reduced, right? So you should be aware of the dehydrogenase group. Okay, and later the alpha ketoglutarate is again converted into succinyl-CoA, where there is a transfer of electrons happening and added from NAD plus to NADH. This NADH is produced. Okay, and here again one more CO2 is released as a waste product. Okay, and here now the succinyl-CoA is converted into succinate, where there is a production of GTP. There is an inorganic phosphate transfer happening, thus there is production of GTP, and here the succinyl-CoA would lose its CoA. Okay, and again this succinate would convert into a fumarate with the production of FADH, where the electrons are added for FAD, producing FADH. Okay, FAD is reduced to FADH. Okay, this is an important step again. Okay, and later in the fumarate is again converted into malate with the usage of one molecule of H2O with the enzyme fumarase. Okay, and here the malate is converted into oxaloacetate, again where NAD plus is completely reduced to NADH with an enzyme called malate dehydrogenase. So if you observe, most of the enzymes in Krebs cycle are all dehydrogenases. Okay, these are all about the electron transports, H plus, how there's a oxidoreduction reactions happening. Okay, so you should be aware of all the names of the enzymes. Okay, now let's discuss about how many molecules are produced, how many FADHs are produced for one molecule of pyruvate reduction. See, so for one cycle of Krebs cycle happening, you have 3 molecules of NADH. See, From conversion into isocitrate to alpha ketoglutarate, you have one NADH produced. Alpha ketoglutarate to succinyl CoA, you have second molecule of NADH reduced. And in the same way, conversion of malate into oxide produces one more molecule of NADH. Thus, for one round of Krebs cycle, okay, for one round of Krebs cycle, you have 3 molecules of NADH produced. In the same way, for one round of Krebs cycle, you have one molecule of FADH produced, converting succinate into fumarate. And in the same way, you have one molecule of GTP produced while converting succinyl CoA into succinate. Okay, and you have 2 molecules of carbon dioxide produced from one round of Krebs cycle, right? So if you think in that way, since glucose is a 6 carbon compound which is produced into 3 carbons of 2 molecules of pyruvate each, this Krebs cycle has to go for 2 rounds of repetition. For one molecule of glucose to get converted, it has to go for 2 rounds of Krebs cycle. And thus, if you go for the net gain, there are 6 molecules of NADH, 2 molecules of FADH, 2 molecules of GTP, and 4 carbons of carbon dioxide produced from one molecule of glucose. So this is how you convert it for every cycle. You should be aware of one single for one cycle, and then you can multiply with how many. Here I have given you the important note of about the normal reaction cycle, that is important. Okay, this is the cycle net gain reaction of the Krebs cycle. Okay, these are the raw materials that are required for the Krebs cycle: acetyl CoA, 3 molecules of NAD, 1 molecule of FAD, 1 molecule of GDP, 1 inorganic phosphate, and 2 molecules of water are important raw materials for your Krebs cycle. And these are the products: 2 molecules of carbon dioxide, 3 molecules of NAD, FADH, GTP, and coenzyme A. I told you coenzyme A is succinyl coenzyme A loses its coenzyme A when it's converted into succinate, and this is a product that is obtained. Okay, this is the complete oxidation, like the complete pyruvate is completely oxidized into the carbon dioxide and NADH and FADH in the Krebs cycle. Okay, so hope you got an idea about the Krebs cycle, what are the steps involved in the Krebs cycle, what is the net gain, okay, and how you are producing the ATP. Okay, now once we are ready with all these FADH, NADH and GTP molecules, we are ready for the electron transport chain would have to happen, okay, which happens in the membrane, mitochondrial membrane. Okay, in the next class I will discuss about it. Hope I have covered all the important points in Krebs cycle. So I have also attached the formation importance of all the enzymes in the link that I have attached in the description. Please go through it. I also important if you are having a little doubt about all these cycles, I have also given you the important videos for this Krebs cycle in the description box. Please check it out. Okay, and see you in the next class with electron transport chain. So thank you, and have a very nice day. Thank you.